Welcome to The Explainer. Today, we're going to decode the machinery of life itself. We're talking everything from the wild strategies of evolution all the way to the intricate web that connects every single living thing. You ready? Let's jump right in.
Okay, let's kick things off with a little puzzle. What on earth could a potato and a sweet potato have in common with a bird's wing and an insect's wing? The answer, and trust me on this, reveals a fundamental secret about how life solves problems. Go on, take a guess. Got it? All right, hold that thought.
And that question, well, it leads us right into our first big idea, evolution's creative solutions. It's all about how nature, when faced with the same problem, can come up with brilliantly similar answers from completely different starting points. It's pretty amazing.
So the key to this whole puzzle comes down to two really important concepts. First, you've got homologous structures. Think about the bones in your arm and the bones in a whale's flipper. They come from a shared ancestor, but they do totally different jobs now. But then you have analogous structures, which are, you guessed it, the complete opposite. And here is the first piece of the answer. Get this. A regular potato is actually a modified stem, but a sweet potato, that's a modified root. So they come from totally different parts of a plant, but they both ended up with the exact same job, storing food. That makes them a perfect example of analogous structures. And this amazing phenomenon has a name. It's called convergent evolution. It's what happens when completely unrelated species independently come up with similar traits, all because they're dealing with similar challenges. And that brings us back to the bird wing and the insect wing. Totally different origins, but both perfectly engineered for flight. It's like nature finding the same genius solution twice. All right, now let's zoom in. I mean, way in. We're going from the huge scale of evolution right down to the microscopic blueprint that makes it all happen, the code of life. So what if I told you that you could take one single strand of DNA and, in just a couple of hours, turn it into over 1 billion copies? I know, it sounds like something straight out of sci-fi, but it's actually a basic tool in biology labs everywhere. This incredible trick is possible thanks to a technique called the polymerase chain reaction, or PCR for short. The easiest way to think about it is like a photocopier for DNA. It works on a really simple principle. In every cycle, it just doubles the amount of DNA. It follows the formula 2 to the power of n, where n is just the number of cycles. So after just 30 rounds, you've gone from 1 copy to more than 1 billion. Incredible, right? But here's a fun fact. While DNA gets all the glory today, it probably wasn't the original star of the show. If we could travel back to the very dawn of life, scientists are pretty sure that its less stable, more reactive cousin RNA was running the entire operation. They even have a name for it, the RNA world. The big breakthrough for this idea came from a completely mind-blowing discovery. Turns out, some RNA molecules, which they called ribozymes, could actually act as enzymes and speed up chemical reactions. Just think about that. It means RNA could store information and do the work. It was a one-molecule show, and yeah, that discovery was so huge it earned a Nobel Prize. You know, this whole approach of trying to understand life by breaking it down into its smallest parts, the DNA, the RNA, the proteins, it's got a name. It's called reductionist biology. And the idea behind it is pretty straightforward. If you want to understand the complex machine, you've got to understand all the little pieces that make it tick. So here's another puzzle. How does the cell cram meters of genetic code into a nucleus that's microscopically tiny? The secret is just some really clever chemistry. DNA has a negative charge, so the cell wraps it around these positively charged proteins called histones, kind of like winding thread around a spool. And where does that positive charge come from? 2 specific amino acids, lysine and arginine. It's a perfect match. Okay, so we've got the code. Now let's see how that code gets put to work inside what is basically a bustling microscopic factory, the cell itself. This is where all the action happens. A great way to picture the Golgi apparatus is to think of it like the cell's internal post office. You have raw materials like proteins arriving from another part of the cell at the cis face. That's the receiving dock. Inside, they get modified, sorted, and packaged up. Then the finished products are shipped out from the trans face, the loading dock, to wherever they need to go. Now, the little machines that actually build the proteins are called ribosomes. And here's a really neat difference between complex organisms like us, eukaryotes, and simpler ones like bacteria, prokaryotes. It's their size, and there's a super handy mnemonic for it. U for eukaryotes is 80S, and pro for prokaryotes is 70S. It's one of those fundamental facts in biology.
Of course, all this activity inside the cell needs a ton of energy. And there's this one enzyme, succinate dehydrogenase, that's a real superstar in the cell's power grid. What makes it so special is that it plays a part in both the Krebs cycle and the electron transport chain, two of the most critical steps in turning a food into usable fuel. It's a real multitasker.
All right, let's zoom back out. We've seen the single cell. Now let's see how trillions of them work together to build probably the most complex machine we know of, the human body. I mean, the development of a human is it's a breathtakingly precise process. After only 1 month, the embryo's heart is already there. By the end of the second month, you've got limbs and fingers. By the end of the first trimester, most of the major organ systems are in place. And by month 5, the first movements are felt. It's like a perfectly timed biological ballet. Just incredible. The heart itself is a marvel. Its rhythm is managed by this really sophisticated 3-part system, and you can remember it with the acronym NAM. The N stands for nodal tissue, that's the heart's own internal pacemaker. The A is for adrenaline, which gives it that emergency boost in a fight or flight situation. And the M is for the medulla oblongata, the command center in the brain that manages the day-to-day rate. Okay, for our final section, let's pull the camera back one last time. We've seen how life is coded and how it's built. Now let's look at how it all interacts on the grandest stage of all, the biosphere. The thing is, no organism is an island. Life is a constant web of relationships. You've got mutualism, where everybody wins. You've got predation, where one wins and one loses. And then there's this really interesting one called commensalism. That's where one organism benefits and the other one is completely unaffected. The classic example is an orchid growing on a mango tree. The orchid gets a nice place to live, and the mango tree honestly couldn't care less.
And the person who really pioneered the study of all these complex interactions right here in India was the brilliant scientist Ramdale Misra. He essentially laid the groundwork for how we understand ecosystems on the subcontinent today. Now, protecting this delicate web of life sometimes means we have to step in. When a species' natural home is in danger, we can use a strategy called ex situ conservation. It literally means off-site, and it's exactly what it sounds like, moving threatened animals and plants to protected places like zoos, botanical gardens, and seed banks to give them a fighting chance.
So we've gone on this incredible journey, right? From a simple genetic code that builds a complex cell to how those cells build an entire organism and how all those organisms create a biosphere. It's all so deeply connected, which leaves us with one final, really deep question to chew on. When we can understand all the parts, all the chemistry, and all the machinery, where exactly is the line between a complex machine and life itself?